So very good evening. So today we are going to see the second part of the general physiology. So in the first part of general physiology, I hope that you all would have gone through the basics of your physiology. That includes the basics of the physiology, that includes the importance of the resting membrane potential. What is a homeostasis? Who coined the term homeostasis? What are the feedback mechanisms that are present in the body? Then we have slowly gone through one important point, that is the junctions, cellular junctions which are present. And then we slowly moved on towards the transport mechanisms which are present in our body. So this is what exactly we saw last time. We ended with one important concept, that is the osmotic shift of water. I probably think that you will all remember. I have told about one important concept, how to represent the osmotic shift of water. So then this is your normal fluid of normal representation of osmosis. And this point is going to be talking about the osmolality. Okay, and this is going to talk about the volume of your ECF, and this is going to talk about the volume of ICF. So this is the exact thing whatever done so far, isn't it? So that point of place, I forgot to mention that this diagram is named as Darrow-Yannat diagram. This diagram is nothing but Darrow-Yannat diagram. Okay, so if at all it is asked in your exam on your viva, you should able to recall this is nothing but your Darrow-Yannat diagram. Okay, so this diagram is the one that is going to help you to differentiate isotonic dehydration, hypertonic dehydration, isotonic fluid administration, hypotonic dehydration. So these four things are represented by this Darrow-Yannat diagram. Now I would like to shortly recall these cellular junctions. Okay, so I think you all remember these cellular junctions plays an important role in maintaining the integrity. This starts from the apex to your base region. So your apex is going to have some, your base is going to have some. So we actually had four important things. I think you can recall that important flow chart: tight junction, adherence junction, desmosomes, hemidesmosomes. I am just having this important diagram here because I want you to remember. Okay, you should not forget. Now coming to the first one, zonular adherence. So zonular adherence is nothing but your tight junctions. So these are calcium dependent. Again, I am repeating. So these are calcium dependent. It is found by E. cadherin. They are linked with the actin cytoskeleton. Mutation in the cadherin will lead to hypotrichosis with juvenile muscular dystrophy. So what will be the symptoms? So what will be the disease which is caused as juvenile muscular dystrophy? Okay, so now coming to this is a autosomal recessive disease. Okay, now coming towards your desmosomes. So this is your desmosomes. So what is this desmosomes? It's present in the macular adherens. So you all know that it is present in the macular adherens. Okay, so the only difference between your zonular adherens and your desmosomes is it is linked with the intermediate filaments. Okay, remember this one point, it is actually going to be having a relationship linked with the intermediate filaments. Right? So the desmosomes are particularly numerous. So the அங்க நீங்க எப்படி கேதரின் பாத்தீங்களோ இங்க வந்து you're going to have a desmosome. Okay, desmoglians and desmocolins. Desmoglians and desmocolins are going to be there. So antibody produced against your desmoglians is going to lead to, you all remember what is that disease? Pemphigus vulgaris. Clear. Next you are going to have is your tight junction. So tight junction is nothing but that is actually zonular occludens. Correct, exactly. Zonular occludens. First one zonular adherence. Second one ocular adherence. Macular adherence. Third one is zonular occludens. Okay. The tight junction is going to play a major role in regulating your epidermal barrier permeability by regulating the paracellular efflux of your water soluble molecule between your adjacent cells. Your tight junctions are also present between the endothelial cells that is going to line the wall of capillaries. So that is the one which is helping you in the formation of blood-brain barriers. So what are the proteins forming? You have three proteins. I already told you this. Claudins, occludins, junctional adhesion molecule. Okay, I call it as jam. So jam eppadi sticky ah iruko adu maari idu sticky ah vechiko ungalku. Tight junction are responsible for the resorption of magnesium ion. Very, very important. Okay, this is not going to allow the paracellular. It is not going to allow your paracellular transport mechanism. Okay, this is not going to allow your paracellular transport mechanism. So here you have your tight junction. So you have your tight junction, then you have your adherence junction, then you have your desmosomes. Okay, so what are the mutations occurring in the tight junction leads to? Number one, it's going to lead to nephrocalcinosis because claudin is the one which is going to get affected, isn't it? So that is going to lead you to nephrocalcinosis, familial hypomagnesemia with hypercalciuria. Hypomagnesemia with hypercalciuria. Okay, then you're going to have this important gap junctions. Gap junctions permit substances to pass through the cell without entering the extracellular fluid. That is the point which you have to understand, the clenching point you have to understand. Clear? The gap junctions are composed entirely of an integral membrane protein called as connexons. Each connexon is going to be made up of 6 proteins, that is your connexins. Okay, 6 individual components which are essential for the molecular transport. Where you are going to find this? You are going to find in the cardiac myocyte, smooth muscle cells. Is it clear up to this? Then you have one important tabular column which you need to remember. Remember, you have different connexins, commonly asked MCQ point of view from your next exam is CX32. What is the CX32? I'll tell you just to 4. Okay, just to 4 I will tell you. CX30, CX31, CX32. Then go for CX40, then go for CX50. 30, 31, 32, 40, 50. Okay, what does your 30 and 31 going to lead to? Just remember this. 30 is going to lead to congenital sensory neural hearing loss. Congenital sensory neural hearing loss. 31 is going to deal with again congenital sensory neural hearing loss. But your 30 is not going to stop there. Your 30 is also going to lead to another C, that is your Clausen's syndrome. Clausen syndrome. That is going to be the other CX30. CX31 is your congenital sensory neural hearing loss. Your 32 is going to deal with X-linked Charcot-Marie tooth disease. Okay, your 32 is going to deal with X-linked Charcot-Marie tooth disease. CX40, connexin 40 is going to deal with idiopathic. Connexin 40 is going to deal with idiopathic atrial fibrillation. It's going to deal with idiopathic atrial fibrillation. And your CX50 is going to deal with cataract. Okay, very simple. 5 connexins you are going to remember. CX30 is going to deal with congenital sensory neural hearing loss and your Clausen syndrome. 31 is going to deal with your congenital sensory neural hearing loss. 32 deals with your X-linked Charcot-Marie tooth disease. 40 deals with your idiopathic atrial fibrillation. 50 deals with your cataract. So simple, isn't it, guys? Yes. So now coming to the last and final one, that is your hemidesmosomes. What is hemidesmosomes? I already told you they are going to be connecting the cells with the underlying basal lamina. That is going to include all the things that is going to be dealing with integrins. It is going to be dealing with intermediate filaments and everything, guys. This is going to be basically connect your basal lamina. This is going to basically connect your basal lamina with your cells. So this has to be very strong. If at all this is not going to be strong, that is going to lead to the disease called as, I think you all know, guys, it is going to be causing bullous femicon. Very simple. That's all. So with this, I just want to go towards the cell structure and function. So before I move towards the body water fluids and other important aspects of general physiology, the very basic for anything is the cell, isn't it? So you need to understand You need to understand small things before you move towards the complicated things, isn't it? You already know what is your cell membrane made of. Phosphatidylcholine, phosphatidylserine, phosphatidylinositol. You have sphingomyelin, you have cardiolipin, that is phosphatidylglycerol. Okay, you have phosphatidylglycerol. So, at this point of time, I would like to emphasize the importance of all these things. You read about phosphatidylcholine, isn't it? Where it is present. It is going to present outside. What is the importance? It is going to be dealing with the pulmonary surfactant, dipomethyl lecithin. What is the LS ratio? I think you all will be reading this LS ratio in your respiratory system. Now what you need to understand is LS ratio is nothing but your lecithin sphingomyelin ratio. Where it is present? This is going to be seen in the amniotic fluid. What is the importance of it? Importance is it is helpful in assessment of fetal lung maturity. So it is going to be important in assessment of fetal lung maturity. Is it clear, guys? Okay, now coming to the phosphatidylserine. Why it is important? Okay, why it is important? Let me tell you. You have this phosphatidylserine. This is going to be an important cell. When this phosphatidylserine is exposed to the outer surface of your cell membrane, that is going to lead to apoptosis. So, when E is going to be exposed outside, that is going to lead to apoptosis. Clear? So, when phosphatidylserine is going to get exposed, that is going to lead to apoptosis of the cell. Okay, the inside to outside shift. Okay, so this inside to outside shift, it is done by scramblase. Okay, so this inside to outside shift is going to be done by scramblase. In a platelet, outside exposure of PS serves as a procoagulant surface, which is going to enhance your blood clotting. Who is going to lead this scramblase from inside your cell to outside? This is going to be done by an enzyme. Okay, an enzyme called as scramblase. When there is a deficiency of scramblase, that is going to be leading to Scott syndrome. So these are the bits and pieces where you should remember at this point of time itself. Why you're going to read that in pathology separately? Isn't it? Very simple point that you can remember one-liner, you can remember now itself. Yes, for scramblase, it's for Scott's disease or Scott's syndrome. Clear. Next, you have this phosphatidylinositol. There is no importance here. Phosphatidylinositol is just a precursor for the second messenger IP3-DAG mechanism. IP3-DAG mechanism is once again an important in the calcium transportation. Clear? That ends the story. Then you have the sphingomyelin, isn't it? What is your sphingomyelin? It's important. Very similar. Sphingomyelin is again involved in your LS ratio, lung maturity, whereas sphingomyelin is abundantly present. Okay, the lipid of your myelin sheath is going to be having this sphingomyelin. This myelin is very important in the saltatory conduction. I'll tell you in the nerve muscle physiology, which is going to be next to this topic. Saltatory conduction, this is required. Therefore, deficiency of sphingomyelinase will lead to Niemann-Pick's disease. Okay, and then your RBC is also going to have a sphingomyelin outside your cell membrane. When there is a deficiency, okay, so when there is an increase in the sphingomyelin content, that becomes acanthocytes, which you can see in the A-beta lipoproteinemia. Now coming to the cardiolipin, I think one of the important, that is going to be your phosphatidylglycerol. What does this phosphatidylglycerol deals with? It is a signature of phospholipid of mitochondria. Why? Because they are abundant inside your mitochondrial membrane. Cardiolipin is involved in entire power generation processes. So when there is going to be a metabolism of cardiolipin, the enzyme which is involved is tafazin. Remember, tafazin is an enzyme that is involved in the metabolism of cardiolipin or your phosphatidylglycerol. isn't it? So the gene that is encoding your tafazin, when it gets mutated, that is going to lead to Barth syndrome. It is going to be characterized by dilated cardiomyopathy, skeletal muscle weakness, growth retardation, and neutropenia. Next, coming towards the anti-cardiolipin antibody. Sometimes what happens is anti-cardiolipin antibody will be formed. That is going to be causing recurrent arterial and venous thrombosis. This is going to be a very important serodiagnostic flocculation test for syphilis. You call it as a VDRL. Very easy, right? When you know the mechanism, you will be reading VDRL test for syphilis. What it is? It is going to be detecting the serodiagnostic flocculation test which is done for your syphilis. Clear, right? Do you have any doubt till this point? Right. You know membrane proteins. So there are 3 types of membrane protein. You have this integral membrane protein, peripheral protein, lipid anchored to the protein. Okay. You have 3 things. Peripheral protein, transmembrane protein, and you're going to have a lipid anchored protein. So each one has some specific importance. If you see integral protein, they are also called as transmembrane protein. Why? Because they are present in both outside and inside your cell membrane. That is why it is called as transmembrane. That is, it is present both in the hydrophobic as well as hydrophilic region. What is the importance of this is transmembrane. You call it as a transmembrane, isn't it? So when you're going to call it as a transmembrane protein, remember guys, when you call it as a transmembrane protein, they are going to be acting as a G protein coupled receptor. Okay. So G protein coupled receptor, they are going to act as a transporter. So 2 roles are there for transmembrane protein. It's going to act as a receptor. It is going to act as a transporter. So you have to remember both these things. Okay. Okay. So this is what your integral protein is. Next, you are going to have this peripheral protein. Remember guys, peripheral protein is very important for the integrity of a cell. Let's have 2 examples for this. When you go for a RBC disorders, you have this hereditary spherocytosis, you have this elliptical spherocytosis. All these are going to cause hemolytic anemia. Okay. For causing hemolytic anemia, there is a structural defect in RBC. This structural defect is due to the protein mutation. What protein mutation? You have 2 important things, ankyrin, spectrin. That is going to cause hereditary spherocytosis, and both this ankyrin and spectrin are going to be your peripheral protein. And also remember, you have this DMD, Duchenne muscular dystrophy. It is going to be caused due to the dystrophy of dystrophin protein, which is absent in your muscle. And your Becker's muscular dystrophy, where dystrophin is absent. No, dystrophin is not absent, but there is an alteration in the amount of dystrophin. When it is completely absent, you call it as Duchenne muscular dystrophy. When there is going to be present but reduced amount, Becker's muscular dystrophy. Everything you will be reading it in the muscle physiology. Now coming back towards the very important thing. Okay, again I am moving back towards a very important concept, that is the cell physiology. Okay, so moving on to talk about the cell physiology. Normally your ECF, ICF concentrations you have to remember. This is what you see in blood. 145 is the sodium mean concentration, potassium 5, calcium 1 to 2, magnesium 1 to 2, 110 millimole for chloride, and 7.4 for pH. ICF concentration is ulta. 140 will be for potassium, 5 to 15 will be for sodium, and calcium will be very 10 to the power of minus 4 millimole per liter. Calcium is going to be around 0.5. Magnesium, chloride is going to be 5 to 15, and pH is going to be 7.2. Clear? Then coming to the cell structure. I'll go one by one. Okay. Talking about the plasma membrane. It is a lipid bilayer. You all know that already. Next comes the very important thing, nucleus of the cell. Nucleus is going to act as a CPU, like a central processing unit. Why? Because it is going to store the genetic information of the body. It is going to be having made up of chromosomes, which carries a blueprint for the individual's characteristics. Each chromosome is going to have a giant DNA wrapped by the histone proteins that is going to form nucleosome. These histone proteins possess usually a high content of basic amino acid, arginine, lysine. So in the complex with DNA and protein, you're going to call it as a chromatin. So why this is important? Because you need to understand this for understanding any cell makeup. Okay. Transcriptionally inactive chromatin is going to be densely packed during your interphase. That is called as heterochromatin. Transcriptionally active irundaduna adu less dense that you call it as euchromatin. So either in this point of time, you need to understand that male will have XY, female will have XS, X and X, isn't it? So among the two X in the female, one X is going to be actively transcripting. So other X is going to remain condensed as a heterochromic clump that is called as bar body, BARR, bar body. Okay, you have to remember this, and that's where you have the disease called as Lyon's disease or Lyon's hypothesis. So what does your Lyon's hypothesis tells us? Heterochromization of X chromosome is a random process. If a single dose of X-linked genes were sufficient, no dimetrial effect will be expected in monosomy of a X chromosome. That is your 45X. But Turner syndrome female has severe somatic and gonadal abnormality. That means that severe somatic and gonadal abnormality, many gene is going to escape the X inactivation, and both X chromosomes are required for the normal oogenesis. Therefore, for a normal oogenesis, you need both X to be normal. Now you need to understand that even though one X is going to be condensed form, even in that case, both X are needed for a proper cell genesis. The membrane of your nucleus is called as nuclear membrane, or you call it as an envelope. They are going to have a special structure called as nuclear pore complex. So it is going to be made of a protein called as nucleoporin, importin, exportin. Import, export. Remember like that. Importin, exportin. Clear, right? Is there any doubt till now? Yeah. So now coming towards the mitochondria. Next you have to remember is mitochondria. Mitochondria, you know that it's called as a powerhouse of the cell. Okay, you would have read this in your 12th standard itself. For your NEET PG, you need UG exam itself, you would have read. But what you need to understand more here is human mitochondria is going to be acting very differently. Okay, there are large number of diseases that are occurring due to the mutation of your mitochondria. You just know that it is a powerhouse of the cell where ATP is produced. But remember, mitochondria is going to be playing a very important role. Apart from your nucleus, mitochondria is the only cell organelle that is going to have own genome. Okay, it is going to have its own genome. Okay, that is why it is very important. It is going to have a separate DNA. Okay, it's going to have a separate DNA. But mitochondria is going to have an ineffective DNA repair system. That is why there is a high chance of mutation of your mitochondria. So, what is importance from this point? Remember, you have various diseases that is going to be causing mitochondrial diseases. Number one is your Leber hereditary optic neuropathy. It is going to cause a painless optic atrophy of optic nerve. That optic nerve is going to get atrophied. That is going to be due to mitochondrial mutation. That is why Leber's hereditary optic neuropathy is important, number one. Number two, you need to understand metabolic syndrome and type 2 diabetic mellitus. Point mutation in the mitochondrial DNA, tRNA encoding your leucine. That is going to cause impaired glucose tolerance and metabolic syndrome. You have mitochondrial encephalomyopathy, lactic acidosis, stroke-like episodes. You have myoclonic epilepsy, that is your seizures, ataxia, increased CSF protein, sensory neural deafness, and dementia. All these things, neuropathy, ataxia, retinitis pigmentosa, NARP, all these things are caused by mitochondrial disease. Why I need to understand this, sir? I'll tell you. Mitochondrial disorders are very easy to treat because artificial electron acceptors like vitamin K3, vitamin C is enough for treatment. Administration of cofactors like riboflavin, carnitine, creatine, that's all. That is going to treat. And oxygen radical scavengers like vitamin E, copper, selenium. This can treat your mitochondrial issues. So you need to understand. Next, you have the very important structure, endoplasmic reticulum. You know that endoplasmic reticulum are of two types, smooth endoplasmic reticulum and rough endoplasmic reticulum. Okay. Rough endoplasmic reticulum is called as granular because it is going to be studded with ribosomes. It is going to be studded with ribosomes. They are involved in the synthesis of proteins, folding proteins. Folding of proteins are done here. So who is going to do this folding of protein? It is done by chaperones, or you call it as heat shock protein. Why it is very important? Because when there is no proper folding, it leads to endoplasmic reticulum associated degradation. Misfolded proteins attacked with a substance called as ubiquitin. When there is a ubiquitin tagged proteins, they are going to be going towards your proteasome for degradation. So automatically your cell is going to get degraded. Unfolded protein, why it is going to occur? Endoplasmic reticulum stress response, you call it. Under certain substances, there will be more production of misfolded protein because there is going to be exporting need in the cytoplasm. When there is an accumulation of misfolded protein, that is going to cause a protective response where it is going to alter the protein in translation. It is going to degrade your misfolded protein and target your misfolded protein for apoptosis. So where you are going to see all these conditions. One of the very common diseases, Alzheimer's disease, where you have this AB peptide tau protein due to neurofibrillary tangles. Parkinson's disease, where you have this Levy body, alpha-synuclein. Prions disease. You will all read this, guys. No problem. In CNS, you will be reading. But then now remember the details. You have Huntington's disease. All these things are going to cause again an issue. Then you have the smooth endoplasmic reticulum. They are going to be called smooth because of the absence of these ribosomes. So what is the importance is here? Steroid hormone synthesis is going to occur. Detoxification of drugs like mainly cytochrome P450 associated drugs. And your muscle cells, specialized SER called as sarcoplasmic reticulum. This is exactly, guys. That's all. Next, coming towards the Golgi apparatus. I think this is going to be one of the boring session in general physiology because fact-based. You need to understand this. Why? To understand the main physiology, you need to understand micro level. You need to understand each and everything in your cell, isn't it? Golgi apparatus. They are going to be involved in sorting of protein. Now, you know nucleus is involved in producing protein, generating protein. Your endoplasmic reticulum is important in folding of protein. What does your Golgi apparatus do? Golgi apparatus is going to do sorting of protein. Like you have who is going to write the letter? Writing a letter is done by nucleus. Then who is going to keep inside the envelope? Endoplasmic reticulum. Who is going to put it in the post? Who is going to write the address? Golgi apparatus. Golgi apparatus is a polarized structure. This Golgi apparatus is prominent in cells specialized for secretion, important for sorting out your proteins. When the Golgi apparatus adds mannose-6-phosphate, it will be delivered to lysosome. When it adds mannose-6-phosphate, okay, which is the enzyme that is phosphotransferase. When there is a deficiency of phosphotransferase, that is going to lead to high cell disease. So here what happens? Your lysosome is not going to function properly. That is lead to accumulation of unwanted substances, degraded substances. That is called as inclusion cells or eye cells. Very, very important, guys. Eye cells, Golgi apparatus. What is eye cells? You need to understand. So this is going to do a post-transitional processing of protein, like adding a carbohydrate group, phosphate group, etc. Okay. Now coming towards the waste bag of your cell, that is your lysosomes. They are called as recycle bin of your cell. They are going to share an important property that all of their are optimal activity at an acid pH. Therefore, acid hydrolysis is the one which is going to occur. They are called as suicidal bag. They are going to be calling as a suicidal bag. So they are going to call it as a suicidal bag. And when any of the lysosomes are deficient, that is going to lead to lysosomal storage disease. Now you know that there are several lysosomal storage diseases. You have 8 important lysosomal storage disorders: gangliosides, Tay-Sachs, Fabry's, Gaucher's, Niemann-Pick, Krabs, Fabry's lipogranulomatosis. Now you can ask me, should I learn this? Of course, this is what you're going to learn in the biochemistry. So whatever you're going to learn in the biochemistry, I'm telling it now itself that G1 gangliosidosis, when there is a deficiency of G1 galactosidase, that is going to lead to G1 gangliosidosis. So here ganglioside GM is going to be a problem. M stands for mental retardation. There is going to be a liver enlargement and skeletal muscle involvement. Death is confirmed by the age of 2 years. Now coming to the Tay-Sachs. You know that Tay-Sachs, very important. This is a repeated asked question in the NEET PG itself. Tay-Sachs is due to exoaminidase A. Exoaminidase A. Okay, exoaminidase A. Why? Because due to ganglioside GM2 deficiency. Okay, so mental retardation will be there. Blindness, death is the age of 3. Then you have a very important Fabry's. So first one, what we saw, GM1 gangliosidosis. There is accumulation of ganglioside GM1 that is going to lead due to the deficiency of GM1 galactosidase. Then you have Tay-Sachs, ganglioside GM2, that is going to be due to the deficiency of exosaminidase A. Now coming to you have a Fabry's disease, that is going to be due to the accumulation of triexosyl ceramide due to the deficiency of galactosidase A. Coming to the fourth one, Sandoff's disease due to deficiency of exosaminidase A and B due to deficiency of ganglioside GM and globoside. Next, you are going to have Gaucher. This Gaucher is going to be due to glucocerebrosidase due to the difference accumulation of glucocerebroside. Then you have this Niemann-Pick. What is this Niemann-Pick going to do? They are going to be due to the accumulation of sphingomyelin due to the deficiency of sphingomyelinase. Then you have the sphingomyelinase deficiency. Then you are going to have a BBE, Krabs disease. This is going to be due to the accumulation of galactocerebrosidase. Galactocerebroside is going to get accumulated. All these things more or less going to involve your nervous system, liver, spleen. That's all. And sometimes your skeletal involvement will be there. Next, you are going to see is a peroxisome. They are microbodies. They are going to involve in 3 vital functions. Number 1, generation and degradation of hydrogen peroxidase. Then number 2, fatty acid metabolism. Oxidation of very long chain fatty acid is going to be occurring in your peroxisome. They are going to be very important synthesis of platelet activating factors and your plasmalogens. You have the detoxification of harmful chemical. Here what you need to understand is there is an enzyme called as luciferase that is going to generate light emitted by fireflies. That is also a peroxisomal enzyme, guys. Okay. What you need to understand here. As I told, all these things are lysosomal storage disorders. Very similarly, peroxisomal storage disorders also there. One important thing is your Refsum's disease. So autosomal recessive disease that encodes for phytanyl-CoA alpha hydroxylase and peroxin 7 receptor protein. You have this adrenoleukodystrophies also. Peroxisomal enzyme catalase, it is used in the treatment of vitiligo, that is called as pseudocatalysis. Okay, now what you need to understand here is, guys, you need to understand how you are going to identify where the disease is lying. Okay, so this is going to be occurring due to the markers I'm going to have. If there is a mitochondrial disease, that is going to have monoamine oxidase, MAO, monoamine oxidase. When there is a lysosomal disease, it is going to release acid phosphatase. When there is a peroxisomal disease, you have the catalase. When there is a Golgi apparatus disease, galactosyl transferase, cytoplasm lactate dehydrogenase. Clear, guys? That's all. This is what we are going to understand. Similarly, for a cell structure to maintain, how your body has bones for maintaining the stature of our body, you have this cytoskeletons that is going to be present for maintaining the stature of the cell. So that is what we call it as a cytoskeleton. So cytoskeleton is going to be very important. Cytoskeleton is maintaining the stature of the cell. So there are three important cytoskeleton. Okay, microfilament, microtubule, intermediate filament. First one is going to be a microtubule. So when you're going to see in the size, microtubule comes first, then comes the intermediate, then comes your microfilaments. Microtubule example is your dynein and your kinesin. So microtubules are made up of two globular protein, alpha, beta. Third protein is also there, that is going to be a gamma. All these things are going to be involved in the production of microtubules. So who is going to be like a microtubule importance? Microtubule, it is going to do a molecular transport. How? By dynein and kinesin. So here your dynein is going to be involving in the retrograde transport or reverse axonal transport. So when this is going to be your axon and dendrite, this is going to be a synaptic terminal and this is going to be your cell body of a neuron. So when there is going to be a retrograde transport from your synaptic terminal to your cell, synaptic terminal to your cell. Okay. So this microtubule is going to be having a antrograde and retrograde transportation by dynein and kinesin. So who is going to cause antrograde? Who is going to cause a retrograde? Dynein is going to cause antrograde. Dynein. And your kinesin is going to cause antrograde. Sorry, kinin is going to cause antrograde and your dynein is going to cause retrograde. This is going to be a kinin. Okay, kinesin. You call it as kinesin. Okay, is it clear? So all these things are going to be a neuronal transporting mechanism. Certain pathology can exploit this retrograde transport to invade your nervous system. Example is your tetanus, rabies, polio, etc. Now coming towards the movement of cilia and flagella. Your cilia tend to occur in large number of your cell surface, helping in the beating activity, which is going to help in the movement. And you know that your respiratory system is going to have a ciliated epithelium. Why? Because to propel the mucus and trapped debris away from your lungs. So the cilia, flagella you can see commonly in the human sperm. All these things are characterized by a axoneme that is going to be comprising a unique arrangement of 9 outer microtubule and 2 inner microtubule. That is 9 plus 2 arrangement, which is interconnected by a dynein. Okay. Whenever there is going to be an absence of this dynein cell, that is your arm, that is going to cause primary ciliary kinesia. One such condition is your Cartnagus syndrome, and you have the cystic fibrosis. Two important diseases you have to understand. Cartnagus syndrome, what happens? It is characterized by a triad of recurrent sinusitis, bronchiectasis, and situs invertus. So primary cilium of your kidney, okay, you have two places, Cartnagus syndrome, that is going to be present. There is a mutation of dynein that is going to cause, number one, primary cilium of your kidney is made up of polycystin, calcium channel ion. Okay. Now what happens? When there is a mutation, that is going to lead to polycystitis or polycystic kidney disease. Okay. Polycystic kidney disease. Then in Cartnagus syndrome, what happens is you will be having a mutation of dynein leads to a ciliary dysfunction, no mucociliary clearance, that is going to cause chronic bronchitis. There is going to be weakening or decrease in the dilatation of bronchiectasis, you have situs invertus, you have male infertility because sperm transportation is going to become difficult. Then you should understand cystic fibrosis, where your CFTR is going to get mutated. So then what happens? There's a loss of mucociliary clearance, going to cause chronic bronchitis, and this is going to worsen, and bronchiectasis is going to occur. Apart from it, you're going to have a pancreatitis, pancreatic stone, gallstones, and salivary duct stone. Mucin is going to be forming a paste, and your CFTR is going to be involving your chloride secretion. So water comes into your lumen by a paracellular pathway. Your epithelium is going to coated with this mucus, and all the foreign body is going to get trapped in that mucus. That is how the entire respiratory system is going to be functioning a mucociliary escalation. When there is a CFTR mutation, you're going to have chronic bronchitis. That's what you have to remember. Now coming to the microfilaments. Guys, before moving towards the microfilament, you need to understand another important, that is your intermediate filament. What happens in your intermediate filament? Remember guys, they are abundant cytoskeletal filaments in your cell. So you have to remember what are the intermediate filaments: keratin, vimentin, desmin. Three important things you have to remember. I'll tell you why. Keratin is going to be a marker of epithelial cells. In a squamous cell carcinoma, tumor marker is your keratin. Vimentin, there are mesenchymal cells. They are located in the mesenchymal cells. So any sarcoma, melanoma, mesenterioma, vimentin will be increased. Then you have this desmin. They are going to be present in the muscles, and any sarcomas, muscle onco diseases, that is going to cause marker. Desmin is going to act as a marker. You have a glial fibrillatory acidic protein, which are going to be located in the astrocytes. They are going to be characterized by presence of cytoplasmic accumulation containing GFAP and heat shock protein called as Rosenthal fibers. So that is going to be there in the Alexander disease. Lamin, again, mutation in the lamin gene leads to progeria. Neurofilament, neurons, they are useful for markers to differentiate neuron from glial cells. Next, going to the microfilament. Remember guys, next you have to go to the microfilament. I'll tell you, microfilament when you're going to consider, remember, they are going to be mainly actin myosin. You will understand the importance of actin myosin when you are going to be discussing about the muscle physiology. Then remember, this Listeria monocytogenes, it's a bacteria that is going to infect your macrophages and cause encephalitis. How they are acting is, they are acting through the cytoplasm of infected cell by the polymerization of actin monomers. So your Listeria is going to have Act A. That is going to be activating the host protein. They are going to cause actin polymerization. That's why this is important. With this, I am completing the entire synopsis, entire thing about the synopsis about the cellular structures. Last and finally, I would like to add upon ribosomes. They are the granule of RNA and protein located as a free form in the cytoplasm or being attached to the endoplasmic reticulum. They are very important for the site of genesis, protein synthesis. They are going to be playing an important role in the protein synthesis. Clear, guys? Okay, with this, so this is your multicellular organism, which is going to have the very big important cell structure, multicellular structure. I think you all understand the various cellular physiology. Now we are going to move towards another important thing. That is going to be your resting membrane potential, which we will be dealing in the next session. I hope you can understand the cellular physiology. That's what the basis of this particular session, cellular physiology. I hope you all understand. Thank you.